je comprends ce qu'ils font, tu vois, mais maintenant, à quel point c'est intéressant, à quel point c'est bien, et pourquoi c'est tout, quoi.
C'est-à-dire, c'est que
Non, ça veut dire, genre, comprendre un peu les maths qu'il y a derrière, quoi. Genre, pas chaque ligne de code qu'ils vont faire, mais comprendre l'esprit. Parce que tu veux devoir, c'est là où tu vas devoir devenir meilleur, tu vois, toute la biologie, tu vas vite facilement, parce que, surtout maintenant, avec la en méthode, ça m'aide beaucoup maintenant. Je vois la progression, quoi. Aussi, sur la théorie et les cortex, tu comprends beaucoup mieux. Moi, quand il y a des cours de maths théorie, je comprends grave pourquoi ça.
ah ouais. C'est là que c'est là que
Quand tu vois une fois, après tu comprends.
ouais, surtout de capter. C'est très bien des têtes de souris là. Mais en fait, on en a pas parlé, mais t'as quand même cité des exemples. Non, c'est demain. Là, il nous prend l'avion. Ah oui, oui, demain.
Okay. Good morning, everyone. A bit early for all of us, I bet. Especially for me. Get everything on. About 2 minutes. Vamos a ver. הגעת, אפשר להתחיל. Okay. Good morning, everyone. We want to start. I just want to sort of get back to where I finished last time, just to make sure that I reiterate the most important points of my first lecture, which have to do with basic computations that are being done already at the level of the retina. So you're all you're all aware of the fact that by now, at the level of the retina, beyond the photoreceptors, the retinal ganglion cells, which send their outputs, have this census around the organization. And we said, okay, when I give this talk to BSc students, I typically mention that, okay, this is to enhance local contrast, to be able to recognize, to enhance the ability to recognize changes in local contrast, which is obviously another way to look at it. But what I mentioned to you is that we can think about this from a computational point of view, is that what's happening is that you have an image, and what the visual system does at the very earliest stage at the retina, at the level of the retinal ganglion cells, is already to do a convolution of the image with a kernel, which looks like a difference of Gaussians or the second derivative, okay? And when you do the second derivative, when you sort of convolve the image, with these filters, that is what you get. And why do we say that this is a convolution function? Because you can think about this at each position in the visual field, there's a cell that has its receptor field at this location, and its receptor field has that property, right? It has this on-center, off-surround, or off-center, on-surround. And we'll talk about the off-center, on-surround in a minute, okay? And when you apply this to, since you have, these array of neurons, each encoding a different position in space, okay? The ultimate result of this is a convolution of your image with this filter, okay? Because this is done in every position in space, okay? So that is the general idea. Now, I mentioned something haphazardly, didn't mean to, but what if we were to look at the, as I said, there's on-center and off-center cells. What would be the output of off-center cells? So first of all, how does this filter look for the off-center? The mirror image of it, right? Okay? So if you apply the mirror image, okay, so think, what is the optimal stimulus for an off-center cell? It's just the opposite of the on-center, right? So you're going to get a mirror image of that. So every place which is black here would be white. Okay, black and white here corresponds to the level of activity. Okay, so they're just a mirror image of what you see for the on-center. Now, good question. Why do we need both? Any ideas? Okay. Didn't expect that would be different. Because you want to sustain, if you are to sustain, let's put it this way. You heard from Idan, I'm sure, that neurons have their spontaneous firing rate, but it's rather low, right? So you don't have much to go below where you are at the spontaneous rate. You have a lot to go up, but very little to go down, okay? So if you wanted to have just as much a response to the mirror image, right, you'd have to keep your sustained response at such a position that you could go as much down as up, right? That means that you need to keep a high firing rate, high sustained firing rate all the time. And that costs energy. So another way to take care of this is to have this on and off pathways, okay, that you sort of, and then in each of them you can go up, you cannot go down, but they are doing the opposite function. Is that clear to everyone? Is it not clear? No, I'll speak in Hebrew. If you don't understand me, just say so, okay? Because I need to know where I'm standing with you. So I think it's okay. So if any point is unclear, please speak up. Okay, you didn't. So I'll go on here. And the other point I wanted to make is that remember that the function here is somewhat similar to a second derivative. Remember that we talked about this? Well, why a second derivative? Why not look at the maximum? Remember we talked about, okay, there's what do we want to detect? A change in the contrast, right? A change in the luminance, sorry, which corresponds to a contrast. So let's say this was X, and this was the illumination here. And I said, okay, you have something like, damn it, let me see it as a function which, okay, so here it would be, this would be black, and this would be white, and you see something like that, I don't know. So you want to detect this, right? There's a big change here in the luminance, okay? Why not look at the first derivative? Okay, what is the first derivative? It's di to dx, okay? And if this is high, okay, let's detect that. But then you'd have to have a setting of when do you say that di to dx is big enough? You'd have to have a threshold, right? If you're looking at the second derivative, once it's 0, you know that you passed. Okay, think about, okay, let's take an example. Since I see some heads here wondering, what am I talking about? Okay, so an easier example. Can you see here? Someone screwed up.
Can we turn the light over there, maybe?
Okay. Okay, it's the other two. Okay, so let's think of a simple example, right? You have something like that. Here's something where you have I very low, and then it goes up. Okay, or let's make it so it's something like that, just so that it'll be more biologically related. Okay, so that's our function of how luminance changes with position. Okay, so what would the first derivative look like? Right, if we apply the first derivative operator, we get something which looks like that, right? Okay, it's maximum here. How about second derivative? Okay, so it's positive here. You can't see. I'll draw it here just for you guys. Okay, so positive here, 0 here, negative here, correct? Okay, so right at the peak here, you'd have something 0 increasing, whoops, correct? Okay, so this here, the 0 crossing corresponds to the maximum here, okay? And the 0 crossing you find irrespective of its height. I mean, if the slope were something like that, you'd also get something, but it would look more something like that, right? The first derivative. The 0 crossing would be at the same position. It would be just something which looks like that. Okay, you guys understand? So the advantage of looking at the 0 crossing of the second derivative is you don't have to determine a threshold. Okay, what's the disadvantage of it? The disadvantage of it is this. Remember, I didn't mean to spend so much time on what we did, but it's important that you fully understand what I was talking about. So, is this. You see here? Okay, so what do we do here? We apply different filters and look at the 0 crossings. Remember? Okay, and we said that if you apply the biggest filter, you're going to, since you're sort of, the biggest filter is spread out, there are going to be fewer 0 crossings. Well, some of the 0 crossings here, okay, are just spurious. They don't have any meaning. We want to get rid, okay? So if you don't have a threshold, of course, then you're going to get all 0 crossings, okay? And then you have to decide, okay, what's a meaningful 0 crossing and what isn't? And you do this by moving from the coarse grain to the fine grain, okay? So that was just sort of a reminder of what we were talking about from a different point of view. Any question? Is that clearer now, or it's just as unclear as it was last time? Because I noticed it was unclear last time. Okay.
I wasn't here on this. Like, which one is the larger filter? The larger filter is the small circle?
No. The largest filter, of course, is the big. Yeah. If you were, I don't expect you to understand this. Okay, that's it. So that sort of was just a reminder of where we stand from last time. And this time, I'm going to talk about, no, that's not the one, sorry. Okay, so that was just a sort of a reminder of what happens till we reach the primary visual cortex. And now I want to give you some, just a sort of a flyover what's happening from in the primary visual cortex, and just do sort of a yeah. Laser. It's even working. Okay, and just give you a brief sort of summary of the physiology, and then we go back to thinking about some computational aspects, okay? So, okay. So those of you who heard the physiology, you can sleep for the next 15, 20 minutes or so, because it's going to be very basic. Okay, but we have to put you in common ground. Okay, so that's the physiology of what you see here is sort of a horizontal slice of the brain. Here's the two eyes, okay? Here's the lateral geniculate nucleus, LGN, which I talked about, which is sort of conveys information from the retina to primary visual cortex. So here in the back is what's called striate cortex or primary visual cortex. Notice a few things. First of all, as I mentioned, on the retina you have a clear mapping, one-to-one mapping between position in space and position on the retina, okay? And a few points to notice, of course, is that if you look at both eyes, most of the visual field is covered by both eyes, right? So you see here the numbers here, 1 to 9, correspond here to the positions 1 to 9, which map where are the receptive fields of these neurons. In this case, the photoreceptors, which transduce light from that position in space. So here 1 maps to here, and 2 maps to here, and so on and so forth. So you see that most numbers here are mapped on both sides. Okay? Now, another point to mention is, okay, that information from the right visual field is conveyed to the left contralateral hemisphere. Okay? So it's conveyed to the left LGN, and from there to the primary visual cortex. So you see that the space here, you can think about this as sort of divided into 2 half faces corresponding to the left and right visual field, mapping to the right and left visual field. Good. And this is just to see, okay, this is true anatomy from post-mortem. So what you can see here is basically, here's what's called the optic chiasm, where there's this crossover. So you see it here, nice and clear. And what you see here, these are all these white fibers here, are the fibers of passage from the LGN to primary visual cortex. So you see here, these fibers of passage corresponding to here. And this is called the optic radiation. So basically, optic nerve, then there's the optic chiasm here. That's called optic tract. And from there on, because you see this spread here, is this optic radiation. Point to mention is when you get to V1, you maintain the nice retinotopy that I was talking about. Okay? So position 1, 2, 3, 4, and 5 here correspond to the positions in space here. So neighboring neurons have neighboring receptor fields. Okay, so retinotopy is a major aspect of representation in the visual system. Okay, that's just to emphasize this. What we see here is basically, okay, another way is the visual field. So fovea, remember, is the 1 to 2 degrees of central visual space, and that there's what's called the foveal magnification, so that, you know, the central 1 degree is represented to a great degree in the cortex. So what you see here is the right and left hemispheres, and you see again the representation that, for instance, position number 2, left visual field is mapped to the right primary visual cortex, and if it's lower field, it's mapped to the upper calcarine sulcus. Okay, so some anatomical information that's not crucial to me for you to know, except for the fact that there's a nice mapping, a clear mapping of the visual field into cortices, and each hemisphere has half of the visual field mapped to it in an orderly fashion. Now, if you move your eye from one position to the other, okay, then it changes accordingly, okay? So it's in retinotopic coordinates, not in world coordinates. We talked about this last time. Okay, and what you see here basically is the same thing. That's, just a nicer representation. Here's the back of the brain, one hemisphere, the left hemisphere here, and you can see where the occipital lobe is, and this is the calcarine sulcus, which you see here, a cross-section of it. So upper calcarine mapping the lower visual field, lower calcarine mapping the upper visual field. Okay, and in fact, you can actually recognize V1 because it has this stripe of genera here. You see, so anatomically, V1, layer forward V1 is very, very condensed, and you can see this here in the layer here. So that's just nice to know, but not really critical. Okay, next. Okay, now the amazing thing is that ever since the First World War, Holmes had the inspiration, I don't know if the inspiration, but the soldiers that he treated had the misfortune to be hurt in war. And what he noticed was, okay, so they had brain damage, okay? And what he noticed was that when he tested them, he saw that, so he tested them just by flashing light at various positions. Okay, so you fixate here, flash light, and you have to detect where is the light and when was it flashed. And what he noticed was that, okay, if they had an injury which was at the lower part, let's look here. You see the nice retinotopy. So you see here the back. Okay, this is back of the brain, front of the brain. So you're looking here at the left hemisphere, okay, and you see it from a mid vision, from a middle point vision. So this is one hemisphere, and you'd have the second one from the other side, and this is the back of the brain. So you see here, what does that map? Well, that maps the fovea. And as you're moving here, look at the dark round circles here. Well, they map the periphery, okay? So there's a clear mapping here, okay, between where the injury was and where the soldier could not locate the dots, okay? And how could he do this? Well, because unfortunately, if you know or you don't know, at World War I, people died from such injuries dramatically because there was no antibiotics, okay? So within a few days they were dead, and then he did the autopsy, and he could see where the injury was, the brain injury was, so he could generate this map, okay? So this idea of retinotopy has been known already for 100 years, okay? When we move to more recent times, in the 70s, Zeki developed a technique to, well, actually, Yuval Weasel started this, but Zeki did this in monkeys, looking at the receptor field. So here's the brain of the monkey. The eye would be here, so that's the back of the brain. And what you see here is just an electrode penetration into, in this case, V1. And what you see now is, okay, here's the fixation point, and what you see here is where are the receptive fields of the cells from number 1 to number 32. So he had many cells that he recorded in this area here, and you can see here the receptive fields. So what do you see? You see, first of all, that nearby neurons have similar receptive fields, overlapping receptive fields to a great degree, and you can predict, as I showed you from Holmes, this was corroborated in Zeki's work, where the receptive field would be given the location in primary visual cortex. So again, the point here, I don't want to waste too much time on this, is that you have nice and clear retinotopy, and there's a greater emphasis, a magnification of the fovea, okay, which serves to we'll talk about it. Why do we need a fovea? But it makes sense, of course. We want to have an area with better resolution, so we can analyze it to a greater extent. Okay, and this is work from Tutel already 30 years ago. Again, just to make the point here, what Tutel used was what's called 2-deoxyglucose mapping. The idea is the following: that if you are activating neurons in any area, they are in need of energy. okay, because action potential costs a lot of energy. And therefore, typically the brain supplies both oxygen and glucose to that area. We'll get to that when we talk about MRI, okay, but this was done, this is just an anatomical technique, invasive anatomical technique that was used on monkeys. And the idea is that instead of you inject into the veins, to the bloodstream, 2-deoxyglucose. What's the difference between glucose and 2-deoxyglucose? 2-deoxyglucose does not change. I mean, typically you break the glucose to get energy from this, but the 2-deoxyglucose is maintained in that format without going into great details. And now what you can do is to label this 2-deoxyglucose with some radioactive labeling. And what you do is, okay, now you inject the 2-deoxyglucose to the bloodstream while the monkey is now watching, or in this case, I think it was, a monkey was watching a specific visual stimulus, such as this one. So what you see is the following. You see this bullseye where the individual pixels here change contrast. So you see this one. I mean, basically, you see this bullseye. And then what he did was he killed the animal. I don't like the word sacrifice. He killed the animal, okay? And then what he does is do a slice of V1 in this case, okay? And so this is a slice at layer 4. And again, without going into great detail, what you can see here, the labeling is such that where you see the darker stripes is where there was more activity, okay? Okay, so there was more radio labeled 2-deoxyglucose, and you can see these nice stripes here, right? And now you can basically map that this stripe here, this ring here corresponds to this stripe here, and so on and so forth. And what do you see here? You see the nice magnification, okay? So this corresponds to the fovea, and this corresponds to the most peripheral ring here. Since this is the left hemisphere, it maps the right visual field, and so on and so forth. So basically, I don't need to spend too much time on it. That was another technique to show the clear retinotopic mapping in V1 in an orderly fashion in monkeys, and basically corroborating Holmes' work and also Zeki's physiological recordings. And basically, this is sort of a key technique that is being used now with functional imaging to map areas in the visual cortex. So I don't know if we have the video here. I'll see in a minute. I think we do. So basically, what you ask subjects in that case is to fixate a dot at the center of the screen, and what you can present is either expanding rings or rotating wedges. Okay, so you have this wedge which is rotating through the from clockwise around the fixation point. Hopefully, I have a demonstration just to show you. So that's nice because that's a technique, basically replicating what you've seen done by Tutel and others without having to kill the human being. Okay, nice to know that you could do this. But you replicate the basic physiology. So that's what I want to show you now. So it's not that interesting, but okay, you have to fixate here, okay? And what you see is these rings which are expanding. And when they're expanding, you map, remember that in the visual cortex, there's two basically axes. There's one which has to do with eccentricity. How far are you from the fixation point? How far is the stimulus from the fixation point? And the other is polar angle. Okay, so if I were to put you now in the MRI, I could predict where your activity would be, and it would be actually quite as you expect. And indeed, just to make the point, so okay, when you present these rings, as you expand them, you expect that activity would change from one area here. So think about it. In one time, you activate one position in let's say, primary visual cortex. And as you're moving, you're activating another position, and this is shifted in time as well. So you'd be able to sort of map this, and here's just the complementary function. Okay, in this case, it's moving counterclockwise. But okay, not that interesting, I must admit. But if you fixate here, then you would get a mapping of polar angle. Okay, and that's complementary. So you can think about this. The mapping in visual cortex is based on two axes, as I said. Polar angle, that's one, and eccentricity, that's the other one that I showed a minute ago. Okay, so you have to do this a few times, and you get a nice mapping. But you have to think of one aspect when you talk about functional imaging, and that is that you're not recording the neurons, okay? It's not direct neuronal activity that you're seeing. Typically, if you've heard from Idan, what is the delay? Let's say that you present a visual stimulus. What's the delay until the cells start firing? Minutes, seconds, milliseconds, whatever. Just give me an order of magnitude, okay? Milliseconds, yes. Okay, the spike is, the spike width is a few milliseconds. The delay is typically not just milliseconds, but tens of milliseconds, depending where you are, right? So, okay, the further you are, the more synapses you have, the more of a delay you're going to get. But that's of the order, okay, in V1, the delay would be maybe 50 milliseconds, and in higher order visual cortex is maybe 100, 120 milliseconds, okay? But look at now the delay of the signal. So that's the MRI signal, and the delay of that signal is of the order of a few seconds, okay? So you're looking at a signal which is sort of delayed, not just delayed, but going up and down very slowly. If you present the visual stimulus here, if you were to record the neurons, they'd be responding right here, okay, in terms of the firing. But this is the MRI signal that you would get. So it's much, first of all, it's spatially, the signal that you see is something of the order of 1 million cells, and it's also temporally a low-pass filter. But still, you can, why did I mention this MRI? First of all, because this is what we do in my lab, one of the things we do. And we have a course that is given by Yuval, the head of the MRI group, was a former student of mine, and myself, here at 2 o'clock. And if you're interested to understand something about the visual system and understand something about MRI, you've missed maybe the first lecture or two, but this is continuing. So come at 2 o'clock. None of you are going to come. But anyway, if not, then you'll hear a little about this from me. So this is what's nice about it is, of course, that this is a non-invasive technique which allows you to map functions in the human brain, okay? And especially if the functions are such that there's an orderly mapping, as we've seen about the retinotopy, okay? So neighboring cells have similar receptor fields. It's not that one cell has a receptor field here and the other away, because then you would not see anything in the MRI signal. But the fact that you have this orderly organization allows you to see something, even if you're looking at a relatively coarse resolution of not 1 neuron or 100 neurons, but rather 1 million neurons. Okay? And that's just the idea. Okay, in the MRI scan, what you do is you would present, say, this wedge which is rotating, and you would look. What you see here in color is where is this wedge in the visual field. Okay, so it's rotating around in a clockwise manner here. And consider if you have a cell here, or it's not one cell here, but now we're talking about 1 million cells. So you can think about the receptor field of these population receptor field of these 1 million cells. Well, they still have a receptor field. It may be much bigger than the individual cell. Probably is, but because of the great degree of overlap between them, as I've shown you before, then you would be able to map this population receptor field. And what do you expect if you were to present this sort of stimulus? Okay, so this is colors here map. Where is this wedge in the visual field? Well, you expect something which is delayed, right? And it has this hemodynamic function which I described before. So your model of response should be something like that, not something like that, because you don't record from the neurons. The neurons would respond automatically or immediately, abruptly, right after the visual stimulus is presented. But here, you're talking about, okay, the x-axis here is of the order of seconds, okay? That's the number of scans, and the scan is, let's say, 1 every second or 2 seconds or something like that. So it would be much more delayed and also following this hemodynamic function. But, and just to show you, okay, here's your model of how the ideal response would be. Here's the response that you get, okay? And what I show you here, just a quick mapping, is, okay, so, and the color here corresponds to where was the wedge in the visual field. So the wedge is traveling, the color, it doesn't change color, but it changes its position in time, right? And what you see here, okay, is, here's an example of, let's say, one voxel somewhere in the orange region here, which corresponds, you see that it's nicely mapping the fact that, okay, it has a specific receptor field, okay? Now, how can you judge how good is this fit to the actual model? Well, once it's written here. So it says cross-correlation, right? So you've heard something about cross-correlation? You know what cross-correlation is? You don't. Okay, so cross-correlation is a function which we often apply. You should be aware of it. Again, just giving the intuition without going into the full math of it. I thought you heard this from, you will, I'm sure you will hear it in your later talks when people talk about responses from monkey, from the monkey. Specifically, okay, you have a model. So let's say the model looks like that. And why is the model looks like that? Basically, because there's the hemodynamic response, and you're giving something, a stimulus which is repetitive, right? It goes, let's say it does a circle. Okay, there's this wedge. It does a circle, goes back to the same position, does it again and again and again. Okay, so that's the time till it gets to the same position. Okay, and then you have the data. So the data would be, I don't know, something much more noisy, but let's say something like that. So on and so forth. Okay, and what you do is you what do we mean by cross-correlating? We're looking at the degree of fit between your model and the data. Okay, so you just match them on a point-by-point basis. Okay, you match them on a point-by-point basis, but when you do a cross-correlation, you can have this in different lags, right? So you can match these like that on a point-by-point basis, and then you take your model and you shift it by one lag, and you do again the match, okay? So what you get is the degree of match that you have between your model and your data as a function of your lag, okay? And then, so going back, that's just the basic intuition of what I'm talking about. Okay, so here's one neuron. If you'd had the same thing and its peak would be at the blue area, then rather than at the yellow area, it would have a different lag. But it would still correspond to the function, to the model function here, right? It would just, you would say, okay, it has a different lag, meaning that it responds to a different position because the lag corresponds here to position in space. You guys are with me? Okay? And therefore, you can use this. This is why we have different colors here, because they map into different lags of optimal matching between your model and your data. Okay. So that's the basic idea here. And just to show you that this is a really nice technique to map V1. This is being developed by Brian Wondell and others. Brian Wondell he's going to be here, by the way, in December. We have a meeting organized. I all urge you to come and hear Brian Wondell. He's really great, okay? But what they've done is exactly what I described, but rather than using the rotating wedge, what you see here is just the expanding ring that we've seen, okay? And now, okay, here's the hemisphere. In this case, right hemisphere, the eye would be here, and this would be the back of the brain. calcarine sulcus is the sulcus in which primary visual cortex is in. Okay, and what you see here is, look here, you see a map here corresponding to, so position 1 corresponds to, you see the red area here is within the red area, that's mapping the fovea. And as you're moving here, it's mapping further and further periphery, going into the eccentricity, right? So this would be eccentric, mapping the blue to violet colors in area 3, and 2 and 1. So notice how 1 is small in terms of its size in the visual field, but it has a great magnification. Okay, that's point number 1. And what you see here is actually, that's just a way to describe this. So signal amplitude is shown here in red. So don't confuse the colors here with here. Just look here, and what you see is the signal amplitude of position number 1. Okay, so you see it's going up and down, periodic. Okay, that's the sign it's periodic because the stimulus is periodic, remember? Okay, and if you look at 3, notice that 3 is lagging compared to 1. Okay, so that's how you can see from here the mapping to here. Are you all with me? Is that clear? Okay, so we get the picture here. So it's a brilliant technique to map the retinopathy in primary visual cortex in a non-invasive way. Very much, why am I presenting this? Because, you know, I'm sure you've heard, if you haven't heard yet, you're going to hear from Elsek how, you know, MRI is just ridiculous, you know, the salmon, the dead salmon in the magnet and all that. You know, okay, fine. First of all, there's answers to everything. I mean, you can bash everything you want, but okay, I'm showing you this because, okay, great. This is a replication of a physiology without having to have people injured with, you know, and being dead after 2 days that you do the autopsy or that you kill a monkey, but rather you replicate the physiology, okay, in a very convincing way, non-invasively in humans, okay? So that's just an existence of proof that this technique is not just bullshit, okay? Now, don't get me wrong, I'm not against monkey research. I've done this myself, and I think it's absolutely crucial. Without it, we would not, I mean, you have to compare this to something, okay? So don't get me wrong that I'm against animal research, but if we can do sophisticated stuff with other techniques and learn something about humans, that's what we really are interested in. So I'm just mentioning this to give you sort of my perspective of this. Okay, here's... okay, enough with the ideology, back to the science. Okay, notice too, but we still have 5 minutes to go. This is just a map that you see for the orthogonal dimension, right? So you can think about, okay, now you can map each position in space with 2 parameters. What are the 2 parameters? Okay, 2 parameters. Okay, let's think usually when we think about this, okay? And then now just usually, okay, for lack of time, here's the fovea. Here's the position in space. How do we usually refer to this? What is the x here and what is the y here, right? But there's another way to look at this. This is called Cartesian coordinates, right? There's another way to look at it. That's polar coordinates. What are we mapping polar coordinates? We map r and theta. Okay? Again, 2 parameters, which allow you to map every position in space using these 2 parameters. So this is what's being used in cortex, r and theta, not x and y. Okay. So here's theta. Yeah, well, 1 minute. I hope I can, I'll explain this and then we'll see. Okay, here's theta, okay? Same area in the brain, okay? In this case, right hemisphere, therefore mapping the left visual field, right? But now we've seen for each position here, point 1, we've seen how does it respond in terms of R, okay? So number 1 is like right at the center of gate, number 3 at the periphery. Now we can look here. Here, we're looking at the orthogonal dimension because each position has 2 parameters, r and theta. This was talking about r, now let's talk about theta. What do we see? we see basically that the colors that are going, so here's the center of the calcine sulcus. As you're going up, okay, this is up and this is down. Going up, your mapping is going from the horizontal meridian to the lower vertical meridian. So this is vertical meridian. Okay, this is lower vertical meridian, and this is upper vertical meridian. This is the horizontal meridian. We're talking about now the right hemisphere, therefore mapping left visual field, right? And you can see here, so the bottom line is as we go here, you're going from the horizontal meridian to the lower vertical meridian, and as you're going here, you're going to upper vertical meridian. And the lines drawn here are the borders between V1 and V2. So in V2, you again have a mapping, a fine mapping of the visual field. It's just a mirror image of what you get in V1. Okay, that's not that critical now, but what I wanted to make the point is that now you can think about each voxel mapping a specific position in space determined by R and theta, okay? So take the information from both of these and you can say where are these cells responding to in visual field. Okay, so just to see that we passed the exam, let's say that if we take here, what would be the receptive field of the neurons whose voxel is here? Okay, so we correspond, let's say, here and here. Any idea? Okay, periphery here, and here the color is purple, deep purple, therefore along the horizontal meridian, okay? So it'd be along the horizontal meridian at the periphery. Correct. What about here? Closer to fovea, right? Mapping the vertical meridian. So maybe something like that. So on and so forth. Okay, I think we need a time for a break. Let's see. Just to finish with the last point here, another way to look at it is to look at it at the following. Here's the map of the eccentricity that I've shown you. One hemisphere, the other has exactly the same map, moving from fovea to periphery, fovea to periphery. So you see red going all the way. This shouldn't be red here. It's a mistake. It should be all the way to purple, deep purple. Okay, and you see that the cortical magnification, and this can also be shown here. So what you see here is the x-axis is eccentricity, and the y-axis is the cortical magnification factor, meaning how much. So you move along the calcine sulcus from here to here. How much should you move so that, let's say, the central degree of the visual field, how much of cortex is devoted to that central degree? Well, quite a bit here, right? So that maps into a great number here. How about the periphery? The peripheral, say, 15 degrees. How much of cortex is devoted to that? Well, much less, right? And this is the function describing the degree of magnification. So, it's strongly declining, meaning that we have a very fine representation of the fovea, very poor representation of the periphery. We'll see what it maps to in terms of the psychophysics in a minute. But I went over time, so let's take. If you don't mind, we'll have a very short break because I need to take my daughter to the doctor at 11. So if we can have like a 7-minute break and we'll start earlier, then thanks. I appreciate that. Yalla, הפסקה. הגודל של הגנגליון צל, או דיברתי על הריספטורים, דיברתי על צריך להפריד בין, האמת שזה נכון שהם קשורים, אבל לא התייחסתי לזה כרגע. יש לך ריספטיפיד יותר גדול ויש לך יותר קטן, על זה אתה מדבר?
זה היה את התמונות שהבנת, את התמונות של הריספטיפיד?
ברור, זה תמיד הריספטיפיד. הגנגליון צל עצמו זה תמונה...
זה תלוי במה, זאת אומרת, איפה היא המיקום של התמונה בתוך ה...
לא, אז הרספטיפיד, אוקיי, יש שני דברים. הרספטיפיד, לא התייחסתי, אבל זה נכון שהרספטיפיד במרכז שדה הראייה הוא יותר קטן, ובפריפריה הוא יותר גדול. אוקיי? אז הרספטיפיד גדל ככל שאתה זז בפריפריה. אוקיי? אבל גם, אוקיי, אז בממוצע.
כן.
הדגש הוא בממוצע.
כן.
אבל בכל אזור ואזור, בוא נגיד, מיפוי שגם של הפוביה, יש תאים הרספטיפיד יותר גדול ויש כאלה עם יותר קטן. והם, כאילו זה מה שאמרתי, שיש כאלה שעושים, הם כולם on center of the arm. אבל בואו נחשוב רגע על ההבדל בין דבר כזה לדבר כזה. כן, אז זה מה שראיתי בקשר שדיברתי על הזירו קרוסינג. זה הקטע שלא היה ברור לך?
כן, הבנתי כאילו מה קורה בפועל, הבנתי רק למה. ואיך זה מתחבר נגיד אם אתה עושה את זה
למה? כי זה פילטר הרבה יותר רחב. זה פילטר יותר רחב.
ואז אומר שאתה לא תראה כאילו שינויים שהם יותר קטנים?
כן, ברור, כי הוא... נכון, מה זה אומר פילטר רחב? פילטר רחב זה שהוא מוריד לך שינויים קטנים. יש לך... עכשיו בוא נסתכל, זה האימג', יש לך דברים כאלה. ואתה מפעיל עליהם פילטר שהוא מאוד, כן, פילטר שהוא נראה ככה, מאוד רחב. אז הוא מוחק אותם, הוא הופך אותם, אתה יודע, כאילו, הוא לא רואה אותם. כי הם לא... אוקיי? טוב, אלה הם כן, הוא כן נראה כי יש פה דברים שהם גם לא פס, זה תדע נמוך. אבל אם היית עושה משהו, נגיד דברים כאלה, ואתה מפעיל עליהם פילטר רחב כזה, כלום, הוא, אתה יודע, מבחינתו הוא לא רואה כלום, כי הדברים האלה בממוצע...
פיוטים ממוצע.
כן, כן, ככל שזה יותר רחב, הוא עושה יותר מיצוע. אז הוא מוריד תדרים נמוכים. אני אדבר על נושא של תדרים בדיוק אחרי ההפסקה. על פוריה טרנספורם, בדיוק, נגיע לזה, כי זה גם קשור לזה. בסדר?
כן, רק השאלה השנייה,
כן.
אמרת שאתה...
רק הלך להביא מים.
סליחה.
לא, לא, בסדר, זה... אפשר לפתוח שם חלון, אין קצת אוויר, נכון? חסר פה, כמו שרק אני נשאר בלי אוויר. יכול להיות שכאילו. אני יכול להבין שזה רק אני. כן, בטח, במודל זה הכל מופיע במודל.
זהו, כי חיפשתי אותו כדי שאני...
ולא ראית? שלחתי למטי, אני לא יודע מה.
כן, כן, נתתי בטח את ה...
כן, שלחתי למטי. Okay. Where were we? Okay. So we were mentioning the magnification, cortical magnification factor. So I hope that's clear to everyone. Okay. So what next? Okay. So I hope, yeah, let's close the door. I think it's quite basic. The point here is you see that there's different lines here. And the lines correspond to different works. Okay, so remember I started with Tutel mentioning the monkey horizontal meridian with using deoxyglucose and vertical meridian also by Tutel. So here are the lines for the monkey. And what you see here is the MRI, fMRI data that I've shown you, basically showing the same function with some variation. Okay, that's human versus monkey, but very, very similar. Okay, so the concept is replicated in humans, and it's also replicated across techniques. Okay, now nice thing to see, okay, you've just seen this function, is to compare it to psychophysics. So I think I mentioned to you that, of course, when we read, we constantly move our eyes, making saccades from one word to the other, because we're unable to resolve something which is in the periphery. And that's just sort of a figure to demonstrate this. So if you fixate here, well, it depends on where you are, okay, you'd easily recognize what these letters here, but if I were to replicate this letter at the periphery, there's no way you would be able to tell this. So this tells you, how much should you increase the size of the letter as a function of its position in the periphery. Now, the further you are, of course, these two letters are not that far away from, in terms of visual angle, so it really depends on where you are seated, but it's something which sort of replicates to some degree the magnification factor, the fact that we are unable to resolve images of the same size as they are moved to the periphery, and that's just because we have fewer neurons to do the job, okay? So it's basically this you can show is sort of replicating the poor resolution that we have in the periphery. Okay. Now this I'll do very, very fast. I'm sure that you will. I'm now till now I talked about the organization, the grand organization of V1 in terms of retinotopy. Next thing I want to talk about in a few, really in a few minutes, is what are the properties of neurons in V1. So I think you all heard this. Correct me if you haven't. That while we were talking about the LGN or the retinal ganglion cells, they have this center surround organization. As you're moving to V1, suddenly you get these neurons which are now contour detectors, edge oriented. Basically, they're sensitive to edges, okay, and to their orientation. Everyone's heard this, right? Who hasn't heard this? Okay, good. Okay, so Yuval and Weisel work, you are familiar with that, so I can really go over this quite fast. Again, here's the 6 layers of cortex. Here's from Yuval and Weisel, the fact that now I'll talk about organization at a finer scale. Okay, retinotopy is at the core scale. This is organization at the finer scale. And that is when they did this and they moved the electrode here and recorded from different neurons, then you find that the neurons are orientation tuned, as I told you. And orientation tuning doesn't change randomly, meaning that, okay, so here the y-axis corresponds to the best orientation of the neuron, and the x-axis here corresponds to the track distance as the electrode moves into V1 here. And the point to notice here is that, okay, neighboring dots here, which correspond to neighboring cells, have similar orientation tuning, okay? what would it be if it weren't the case, if it were completely random? You'd see that the dots here along the x-axis are spread just randomly, right? While you see here that neighboring dots are quite close to one another in the y-axis, okay? So I think you're familiar with the idea of orientation columns. Anyone hasn't heard the term orientation column, doesn't know what it is? Okay, so that's just proof of orientation column, the fact that neighboring neurons have similar orientations. Okay, and here's the Hubel and Wiesel work. If you're all familiar with that, I allow myself to move forward with this. Okay, so just to mention that if you go normal to the surface of cortex from layer 1 to layer 6 here, you find that all cells have the similar orientation preference, except the ones in layer 4C, which don't have any preference whatsoever. Okay? If you do a tangential sort of oblique penetration, then you replicate what I showed here. Neighboring neurons have similar orientations, but it's changing as you're moving along the electrode track, as you see here. Okay, good. And that led Hubel and Wiesel to make the point that cortex V1 is organized at the micro scale such that neighbor neurons have across the layers have a similar orientation. This was verified, corroborated 10 years later or 15 years later by another technique called optical imaging. The nice thing about that technique is that now you open a chamber to the brain. Basically, without going into great detail, what you do is you have a camera that is, and what you use is sort of a light voltage sensitive indicator, which basically converts voltage, electrical activity, into light. Okay? And that allows you, now you have a camera that you can measure the light, and you present to the animal, the monkey in this case, a stimulus, let's say a vertical grid, and you get an image display. So this is what you see from the camera here. And now you can start, the colors here don't mean, I mean, the colors, let's say red means high activation and yellow means low activation. And you can now present these images at different orientations. Okay, so it could be vertical grid, horizontal grid, and so on. So these are the numbers here, and you get a different pattern of activity. Okay, so let's say that the whites here correspond to high activity, black is low activity for a given grading. Now, if you give a different grading, you get a different pattern of activity. And the nice thing about it, why am I mentioning this, is that here, now this window here of the order of 1 centimeter by 2 centimeters or something like that. So you don't see just one cell, another cell, and so on, as Hubel and Wiesel have shown. But here you see the picture from, millions of cells together. And you still see this map, okay? Why? Because there's a mapping of orientation, nice mapping of orientation. And basically, if you superimpose these maps here, you get something like that. That tells you what orientation is preferred in a specific region of cortex. Okay? And that's shown here. So that's just a replication of what we talked about, the mapping cortical columns here. So you see that this has some grain to it. It's not random. Okay? So the colors here correspond to the orientation of the specific stimulus, the preferred orientation of the specific stimulus. And let's look here. So here's an area which is prefers that orientation here. Now, and I point here is that you see these what's called pinwheels. So you see areas in which these converge, like here. Okay, so the orientation changes dramatically, very fast. While in others, you see here the area is yellow, meaning that the orientation is rather steady for a given orientation. I don't know, that orientation. So there's some structure to that. Again, the details here don't really matter, except what's nice about it. Why am I mentioning it? Because it's a different technique that corroborates previous findings, again, showing that there's these nice orientation columns, and you can sort of map the preferred orientation here according to color, and the contour lines here correspond to changes in the orientation. Okay, great. Another point in terms of organization of V1, visual cortex, is that you don't just have the orientation, but you also have eye of dominance. You've heard this? Anyone has not heard about eye of dominance? Eye of dominance is which eye is more dominant? Is it to a given area in primary visual cortex? Is it the left eye or the right eye? So that's, again, work that has been done by Hubel and Wiesel. They've moved an electrode into primary visual cortex and moved here along the electrode track here. And what you see is here is cell number 1, cell number 2, cell number 3, so on and so forth. And what do you see here? You see along on the y-axis the ocular dominance group. I'll mention this in a minute. And in the x-axis, distance along the electrode track. This was the first cell, second cell, so on and so forth. And what do you see? When we talk about ocular dominance, okay, which one eye is more dominant? So I can present the stimulus at the receptor field of a cell, once covering one eye, then covering another eye. Okay? If it's responsive only to one eye, let's say to the left eye, okay, in this case, there's no input coming from the right eye because you cover it. Okay? So it's only from the left eye. Okay? So if it responds strong to the left eye, we would just to the left eye, we would call it category number 1. If it's responding only to the right eye, we call it category number 7. If it's responding equally to the left eye and the right eye, we call it category number 4. What do you see here? That this is changing. Again, not abruptly, not randomly, but in some orderly fashion. Meaning that there's, okay, if you think about it, you can think that there's an area of dominance of the right eye, then an area of dominance of the left eye. So left eye, then right eye, right eye, and so on and so forth. So this is the other parameter. If we were talking at the micro scale about orientation, the other relevant parameter in terms of organization of V1 is ocular dominance. Okay? And in layer 4, 4C, this ocular dominance is complete. Okay? The cells respond either to the right eye or to the left eye. Okay? If you move away from layer 4 to other layers, then the ocular dominance is not complete. Okay? It's a preference to one versus the other. Now, layer 4C is the one that's getting its information from the LGN, and that's just shown here. Okay? So think about it. Okay. At the retina, if you record from a cell at the retina, left retina, okay, is it going to respond to when you close the left eye and the stimulus is just shown to the right eye? Of course not, right? Because it's on a retina. It's eye dependent. That's obvious, right? Now, interestingly enough, if you move to the LGN, you still maintain this preference to, not preference, but complete preference to either one eye or the other. We said, notice here, that if you look at, let's say, look here at the, what is it, the right hemisphere, it gets information from both eyes, right? You see, through the chiasma from the left eye and not through the chiasma from the right eye. Well, how do you maintain this ocular preference? Because the cells, the information terminates at different layers. Okay, so if you see here, see 1 to 6, there's 6 layers here at the LGN, and the information from the contralateral eye terminates at the layers 1, 4, and 6, and from the ipsilateral eyes, it's 2, 3, and 5. Now again, I don't expect you to remember which layer does it terminate, but the point is that it's segregated. Okay, and it's segregated in the LGN, and as you move to the striate cortex, when you get to layer 4, that's the input arriving from the LGN. So the next station from the LGN is layer 4 of V1. There, again, it's still segregated. But as you're moving out of layer 4, now the input in layer 3 or layer 5, outside layer 4, is now from both eyes. Okay? So that's where it now becomes integrated. Input from both eyes is integrated. Okay? And that's shown here. Remember that we talked about to tell. That's another way to look at it. You look at the ocular dominance columns using the deoxyglucose technique that I mentioned, that you see these stripes. The stripes are because you've sort of given the stimulus only to one eye. Okay? So you see a nice stripe here because The black areas are the ones that are responding to the eye that was stimulated. Okay, so you see this nice correspondence. At the level of layer 4, if you were to do this in other layers, you would not see this, because now the information is integrated. Yes?
The meaning is that in layer 4 we get a 3D image or not?
No. Okay. Close your eye. Do you see me? Can you tell if I'm closer to you than the other people here? Of course you can, right? You're right that we use the two eyes to infer some information about 3D, but you don't need the two eyes in order to do this, okay? So it's not, I answered you in a tricky way. I'm saying that in order to extract 3D, stereopsis is a cue, and stereopsis is based on the information, comparison of information between the two eyes, but it's not the only cue, certainly not the strongest cue. For instance, occlusion is a very important cue. Motion parallax, the fact that I'm moving, if you sit on a train, look outside, the things that are further away are moving slower, the things that are closer to you are moving faster, right? So there's a lot of cues for depth, not just disparity. Anyway, going back to this, so just to summarize, we have in V1 this organization where you have a map of orientation preference, orientation columns, and you have eye of dominance being contralateral, ipsilateral, and the eye of dominance here is shown here in the eye being ipsilateral, C being contralateral. So you see that, and the white sort of lines here correspond to the orientation preference. So you see that both parameters are mapped in an orthogonal way. So all of these would be neurons here corresponding who have a preference to the contralateral eye, and they would have a different orientation preference depending on these contour lines that are here. Okay, and that's just a lot of anatomy and detail. No, these are important details, but I want to have time to mention a few other things. And okay, one other thing that I want to mention, and as I said, you see this very clearly in layer 4, okay? Because that's where the input comes from the LGN, okay? So here, and just to complicate things, the anatomists make our lives very complicated. They're not just talking about layer 4, but they divide layer 4 into 4A, B, C, alpha, C, Don't get there, but okay. So most of the input goes to 4C, layer 4C. So this is where you'd see this nice and clear difference between the contralateral eye and ipsilateral eye, most clear in layer 4C of V1. Okay, and that's just to show you. Okay, sort of the input arriving from the LGN, mainly arriving to layer 4, and then there's the output from layer 4 going to layer 2 and 3, also to layer 5 and 6. Layer 6 is going back to LGN. Remember, we talked about feedback here. Layer 2 and 3 to other cortical areas. It's quite complicated, but you get some idea of the interconnectivity within visual cortex. I think you're going to hear something about this from Idan, okay? So if you haven't yet. Okay, just sort of an interesting point. Yuval and Wiesel got their Nobel Prize in 1981, I think, for their work on, deciphering the organization of primary visual cortex, the orientation columns, the developmental aspects, critical period, and so on and so forth. They missed something very big. Remember that when we talked, I showed you an example. Let's just see this. Here. Okay, here's the penetration that they've done. See, they're going from layer 1 to 2, 3, and 4. You see, okay, there's these cells that are mapping orientation. Well, that's from their own work here. Now, what was found out years later by Yuval and Marge Livingstone is, okay, that's not the whole story. And why is it not the whole story? You could think about it. Okay, something was neglected. I mentioned this last time. We talked about color vision a bit, right? Talked about the photoreceptors being wavelength sensitive. How do you infer color from the vector of these three photoreceptor response? Well, what about color information? we talked about orientation, very nice for contours and everything, but what about color information? Well, apparently, you know, in the early 1980s, they found that there's some technique in which you can get these, you see these darker blobs here? These sort of cytochrome oxidase blobs. Not getting into the biochemistry of this at all, but if you do this staining, you can get this nice blobs. Don't pay attention to these white elephants. They're not the relevant ones. Look at the blobs here. And the point I'm trying to make is that if you now, once they notice these blobs, they try to record from the cells in these blobs, and they found out that they were not sensitive to orientation, but rather they were color sensitive, okay? So if we were talking about cells which are on-center or off-center, we talked about these. There's also, of course, these cells also in the LGN, which are color sensitive. So they prefer red in the center, green in the periphery, or green in the center, red in the periphery. Okay, so they do sort of they are chrominance contrast sensitive rather than luminance contrast sensitive. Okay, and that's just to show you. Okay, here's a picture here. If you are to look at differences in luminance, okay, so there's a big difference between here, right? So here's if you sort of plot the luminance level of the different flowers here, and this is if you sort of you ignore the luminance. You don't care about the luminance. You care about the chrominance, and you get that picture. Well, of course, that also is informative to know something about the world. It's not just the luminance that we use. We use also the chrominance, okay? And these cells are the ones which are encoding chrominance contrast. Kenda.
These cells don't get information like other kind of information, like we talked before about orientation, for example, just about color. There is no overlap between information.
So this is a different set of cells which are chrominance sensitive. Okay, so there's a different pathway. Basically, didn't get into that, but there's the magno and the parvo pathway. I may have mentioned this regarding the LGN. So we're talking about two pathways, and basically the parvo pathway carries information about chrominance. Okay, so it's in different layers of the LGN in the parvo. Yes. Okay. And so the more recent picture from the late 1980s is that you also have these blobs here which are color sensitive, here corresponding to the blob that you see here. Okay, so they're sort of, and they typically are found in layer 2, 3, or 5, 6, not in layer 4. Okay, so, but that's just why am I mentioning it? Because it's nice. I mean, even these Nobel laureates, they must have noticed that as they're moving their electrode, and each time they're blind. They don't see the cells, right? It's not like the techniques that we have nowadays. They were sort of blind moving their electrode, and each time they get closer to a neuron, they see an action potential. And then typically, when you get the action potential is just spontaneous rating. It's spontaneous firing of this neuron. And what you do next is just now try to map the receptive field and find what are the optimal stimuli. What is the optimal stimulus for that specific cell? Okay. Now, it is obvious that they have. It's obvious to me. I don't have any evidence that they must have encountered such cells as they were moving through the blob. From time to time, they were going through a blob. Okay. And there they would not find these orientation selective cells, but they don't report it. Okay? They don't report it. It's only 10, 15 years later that, you know, this has been mentioned. Okay? And they got the Nobel Prize for a good reason. Okay? But just shows you that, okay, you know, people, why they tend to select what they present. I'm not saying that they're cheating or anything like that. I'm just saying that that we are all have our biases to, okay, how do we perceive what the organization is, and then we sort of we have to be aware of it. That's the main reason why I'm mentioning this, not just the fact that, okay, color is also an interesting factor which has been neglected in previously. Okay, so here's the again, remember this slide that I've shown you, that, okay, here's the original input, and you can think about, you know, at the level of the rods and cones, the analysis that's done, and different photoreceptors here, and the LGN cells, which are doing Okay, that's the output to the LGN already centers around, okay, and that would be sort of a convolution with this difference of Gaussian, the Mexican hat receptor field property of cells at the retinal ganglion level, which correspond to this here, and this is the same when we're talking about color commonness, right? Color difference, okay? So yellow to blue or red to green, okay? And then we get to visual cortex, and now we're going to talk a bit about the sort of functions that are done in visual cortex. So we talked about the fact that, okay, we have orientation selective cells, right? Specific to a specific orientation. Let's get, see, this is displayed to some extent here. If you notice that there's more, okay, the orientation, the ones which are more selective to, I don't know if you can see this, but compare this to this, okay? So this is to a vertical orientation filter. This is to a horizontal orientation filter, right? So you see the difference here between the two images corresponding to the function in V1. Okay, so a few words about this. Maybe 10 minutes or so to get into this, maybe 15. Okay, so when we talk about V1 cells, they're orientation selective. Here's a few examples here. Here's another and another. So this one aspect is what is the angle of orientation, but another is, if you want, what is first of all, the size of the receptor field and the size of the bar that gives the best response. Okay, now is it a bar? Well, sometimes you find this is typical of neurons in V1. They prefer a bar at the center and something which is the opposite, dark bars neighboring it, okay? You can think about this in terms of spatial frequency. Well, we'll mention this in a minute, but here's just a mapping of these. These neurons could be mapped into sort of a polar mapping again. Center would be, okay, so the radius here would correspond to the spatial frequency. Okay, so how many cycles, let's say, cycles do we have within a visual degree? Okay, so the greater, that would mean many more bars in this receptor field. Okay, so 16 bars in the receptor field versus, I don't know, 1 bar within this receptor field of 1 degree. And so that's one aspect. And the other, which is mapped here by the angle here, would be the orientation. Okay, so this would correspond to an orientation, I don't know, of 90 degrees, and this would correspond to 180 degrees. Why do we have something which is at 360 rather than 180 degrees if we're talking about orientation? Because it may be direction selective. Maybe if you move these bars to this direction, we'll call it 0. If you move you present the stimulus inside the receptor field. Okay, you find what is the best orientation. This one. You find what is the best spatial frequency. This one. Okay, that's medium spatial frequency. That's higher spatial frequency. That's lower spatial frequency. You're all with me? So far, so good. Now, that you'd expect. Okay, orientation changes from 0 to 180, right? Think about it. But why do we have here a map of 360? Because it may prefer motion upwards, and then we'll call it 0, versus motion downwards, we will call it 180 degrees, okay? So in that case, there's 360 degrees here. Okay, that's not very critical, but it makes the point that what you see here now is different receptor fields of different neurons here. So this one corresponds to, it prefers, I don't know, a specific spatial frequency, say 2 cycles per degree, at a given orientation, okay? If we were to place it here, it would be the same orientation, same spatial frequency, but opposite direction of motion, okay? So often the cells are also selective to directional motion. Okay, so neurons in V1, you can think about them as selective to orientation and spatial frequency, or bar length, okay? Now this is a major issue here, okay? And this is relating this to what I started talking previously. You can think about the visual image. How do you map the visual image? Okay, there's a visual image. There's 2 ways to map it. Okay, you can say, okay, what is the level of intensity at different positions in visual space, in X and Y, or in R and theta. But another way to look at it is from the Fourier, from the Fourier transform. So what you're looking at, you have a visual image, and you can decompose it into sinusoids at different frequencies, okay, and at two different directions, okay, U and V, okay. So look at this one. This is a sinusoid, vertical sinusoid. This one is the same. It's just a horizontal sinusoid, okay. And you can think that you can take an image and decompose it into different sinusoids and see how much energy you have in each of these sinusoids. That's tricky. Why? Because we're talking here about a two-dimensional case. Let's think about a one-dimensional case. Open the light here and I'll make it clearer. It's not trivial to understand, okay, you have these bunch of sinusoids here. How do you map a visual image? What am I saying? You can take a visual image and say, okay, how much energy do I have in each of these filters? And that would correspond to your image. Okay? Now, who here has not heard anything about the Fourier transform? Okay, heard Fourier transform. Who here knows what a Fourier transform is? Okay, thinks he knows what a Fourier transform is. So you'll help me now, because, okay, now this is what we're going to talk about in the next few minutes. Okay, so the idea is the following. Let's take an image, which is, just to make it simpler, one-dimensional image, okay? How can I draw it? I can draw it X here, just as I've done before. This would be intensity, and you'll have something like that. I don't know. Okay? It could also be a face, okay? Same thing. That's one image, that's another image here, okay? Now, you may have heard this from Eli in the auditory domain. Okay, you can take this and now decompose it. How can you code it? You can say, okay, what is the value, the intensity value, at each position in space? That corresponds to that specific image. But another way to look at it is from the frequency domain. And then you can say, okay, let's look at it. We try to decompose this image or that image into how much energy it has using different sinusoids. Okay, so you have, let's say, a sinusoid which looks like this, and a sinusoid which looks like this, and so on and so forth. Okay, and the idea is, and that's not, it's, I think, non-intuitive to figure out that you can take a group of these sinusoids, okay, and build any image you want. That's the principle of the Fourier decomposition. That as you can map this image using, okay, what is the luminance at every point here at X, you can do the same using these sinusoids, okay? That's non-trivial, but that's easily, that can be proven mathematically. Okay, now take this for granted. I don't have time to get into this, but let's just give a sort of a simple example. So here we are talking about the two-dimensional case, so it's a little more tricky to think about it. But think about, okay, going back to here, the one-dimensional case is that you talk about these, right? So this is here. What does that correspond to? That's the mean luminance of your stimulus, okay? No modulation. Here you're talking about one cycle, two cycles, so on and so forth, right? So you see that as you're moving here, the frequency is becoming larger. And now you're asking yourself, okay, you have an image. How much of the image can be explained by the mean luminance? How much can be explained by one cycle in your image, two cycles, so on and so forth? Okay, so that's the basic idea. Let's look at what the next stage is, okay, remember what I was talking about V1? Well, in V1, you have these receptive fields here, okay? So they're orientation tuned. There's a mapping to all orientations here, but there's also spatial frequency tuning, okay? So as you move here, okay, the receptive field becomes bigger, and it maps a lower spatial frequency compared to this is high spatial frequency, this is lower spatial frequency. You can think about this here, okay? You see here, what you see is basically sort of a mapping Each circle here corresponds to a receptive field of neurons. So you have a sort of a mapping of the visual field, and the receptive fields are becoming bigger as you're moving out to the periphery. And another thing that you need to be aware of is that there's two types of cells in V1. One is called a simple cell, and one's a complex cell. A simple cell has that sort of receptive field, okay? And it responds to, say, in this case, a bar at 45 degrees, but it prefers a dark bar in the center and next to it 2 light bars, okay? It does not respond if you present a light bar at the center and 2 dark bars by its side, but it does respond very well if you present a dark bar and 2 light bars by its side. A complex cell is also found in V1, and basically the idea is that it gets information from sums the information from simple cells such that it's phase insensitive. So it responds just as much to a dark bar next to a light bar than to a light bar next to a dark bar. So it responds to both of these structures here, and this can be seen here as you're moving, increasing contrast, either when there's a dark bar and a light bar, or a light bar and dark bar, both lead to a response. Great, a lot of details here. Sorry about this. Okay, so there's simple cell, complex cell, and there's sensitivity to orientation and spatial frequency. Great, okay, what do we make of this? All of this together. Well, okay, here's just another example showing that these are cells that are recorded from V1, so they are sensitive to spatial frequencies, as we've seen before. No information about the orientation in this case, but you see that the different cells have different sensitivity to different spatial frequencies, and here the y-axis is contrast sensitivity. So the higher it is, it means that they have a greater sensitivity, okay? So these ones are this cell here is very sensitive, these ones are poor. But you see that they're sensitive to specific spatial frequencies, as I've mentioned before. Now, why am I mentioning this? Because so are we, okay? So if I present you a stimulus which looks like this, now, if you notice, what do you see? You see nice bars here of light and dark, but as you're moving up here, you might lose them, okay? And here, I'm sure that here no one sees anything, right? Now, if you notice here, what do we see? Again, depending on the distance, I would say that you see here, okay, you're losing it somewhere here. But as I'm moving here to higher spatial, so these would be corresponding to low spatial frequencies, and these are high spatial frequencies, right? Here, at least from my distance, I can resolve the contrast till here maybe, okay? And I'm sort of plotting where I'm losing it. Here I certainly don't see anything, I bet you don't, okay? So we have some function here which is called the contrast sensitivity function, okay? And this is something which is somewhat something like that. Okay. Now, cool psychophysics, okay? And related to the Fourier transform. Let me explain. So let's say that you're presenting this sort of stimulus, okay? And now you have to, okay, so this is in a given spatial frequency here, and you're looking at this for quite, let's say, a few seconds, and now I present a different stimulus, and you have to tell, okay, do you see a grating here, or there's nothing there, okay? Well, what you see in the black dots here are just what I showed you before. This is your sensitivity as a function of the spatial frequency. So typically, as I've shown you, here, at some point you cannot resolve the lines here, right? You lose it at high spatial frequency, and also drops in low spatial frequency. Corresponds to this picture that you get here. So higher sensitivity, meaning that you can tell this at low contrast, okay? Now, if you adapt to this stimulus, and I do this again, you get a change in your contrast sensitivity. You lose sensitivity at this area here, but it's limited to a specific spatial frequency. It doesn't affect your performance here or here. It affects it only around your adapting stimulus, okay? And here, if you take the difference here, you get here. So you see here that there's sort of it suggests that there's a spatial frequency channel. Okay, there's a channel of a given bandwidth here, which is just the effect that you get by adapting that to that stimulus. If you were now to use a different spatial frequency, what would you get? Let's say you would be using a much higher spatial frequency. You'd be getting a hump here, a loss of resolution, or a loss of an increase in the threshold, loss in sensitivity, which would be, again, specific to your adapting spatial frequency.
What did you change in the second stimulus?
What did I change here? See, I'm presenting different stimuli of different spatial frequencies, and you have to tell when do you see it, okay? And what you see in the open triangles here are the cases in which after adaptation. So you did this before adaptation, now you present this as high contrast, say for a minute, and then we check how well do you do after that adaptation, okay? And you lose your ability, but it's just limited to the adapting stimulus, within the bandwidth of the adapting stimulus. Now, okay, now here's the point I want to make about the comparison between a square wave and a sine wave. So you can use the adapting stimulus once you use a square wave stimulus, and once you use a sine wave. A square wave is where the changes in luminance are abrupt. A sine, of course, is something, okay, a sine wave. And now, so this was done by Campbell and Robson in 1968, already 50 years ago. And look what they show here. They show, okay, now the contrast sensitivity when you're using a square wave grating versus a sine wave grating. So the triangle is the sine wave grating, sorry, the square wave grating, and the circle is the sine wave grating. So there's a bit difference. There's somewhat of a difference. Notice that the triangles are always above the circles. Okay. The question is why? Okay, so you're adapting now, and you're asking, in this case, you're measuring contrast sensitivity. Sorry, this is without adaptation. Take it back. You're using, let me explain what the procedure is. You're presenting this. You're presenting a stimulus. It's either a square wave or a sine wave. Okay? And now you have to tell, did you see something changing, a modulation or not? Okay, so it's a detection. Okay? And now you ask yourself, okay, what is the contrast, or what is the sensitivity in which you can detect this? And you see that there's a difference. You typically do here better than here. Okay? Now why? The question is why? And that has to do with the Fourier analysis. Okay, so I'm getting back to the Fourier analysis. I'm losing time. So I will certainly return to this on my next talk, but let me just leave you with the intuition, or at least some of the intuition. And the intuition is the following. Remember that we talked about Fourier analysis? Well, let's take a square wave of a given frequency. Now we want, okay, this is an image, right? It's dark in some areas, light in some areas, just as I've shown you here. I want to decompose it in frequency space. How do I decompose it? Okay, it has a fundamental frequency. So you've heard something probably in the auditory domain about, spectral decomposition here. Okay, it's exactly what we've done before. What is the basic frequency? Well, it would be the sinusoid fitting this, right? So it would be something, I don't know if I'm doing this ideally, but something like that, right? So that's the fundamental frequency of this one. But it's still not a square wave. I need other frequencies as well. Okay, so this would be 1F. This would be the fundamental frequency, or F. Now, if you do a Fourier decomposition, the next one would be 3F. 3F would have 3 cycles in this, okay? So it would look something like that. And if you sum these together, what you're going to get is something which looks like that. Okay? So that's closer to a square wave than originally was, okay, with just using f, okay? But of course, the amplitude of 3f should be smaller than the amplitude of f, okay? So 3f is smaller in amplitude than f, and one can show that also you have a component of 5f, and this is called the Fourier series. You can show mathematically that you can build a square wave from a sinusoid function of f. Okay, so you have f plus 3f plus 5f and so on, plus all the odd ones. But the amplitude of f would be 1. The amplitude of 3f would be a third of the amplitude of f, and so on and so forth. Okay, so the amplitude is decreasing. Just as I've shown you, it's clear that you want the amplitude of 3f to be smaller than f. Okay, why is it exactly a third? So it doesn't really matter. But the amplitude is decreasing in its effect. But there is still a component of 3f. Okay. Now think about it. Okay. Now if we go back to here, that's where we will stop. Two things here. First of all, what would be, think about this. Okay, so let's think of f, just f. Okay, so here's the square wave, and now I'm plotting f on top of that. Right? What is the amplitude of f compared to the square wave? Should be bigger. It's actually bigger, okay? And the okay, so that's the amplitude of the square wave here, and that's the amplitude of the fundamental frequency. It's bigger, and it's bigger by a factor of 4 over pi, which is a little, okay? 4 over 3.4, okay? It's a little more than 1, okay? Okay? So, take that. We'll stop here, but okay. Now, you notice that the triangles are always higher than the circles. And I'm saying it has to do with this ratio here, okay? I'm saying that, okay, now when you're using this stimulus rather than this stimulus, the amplitude of this one is bigger than that. And the difference here corresponds here to 4 over pi. So you see here all the points here, they correspond to something which is higher than 1, 4 over pi. Understand? I didn't have time to go into this. I will get it back into it in our next meeting, because I think it's a very important point. Just to sum up, what I want to make, the point I wanted to make is, okay, we have evidence for Fourier decomposition taking place in V1. Okay, cells are sensitive to the spatial frequency, not just orientation, but spatial frequency, and we can understand some of the psychophysics. Okay, and the single unit recordings as such a function. Okay, now we're going to talk about as edge detection, or in Fourier space is spatial frequency sensitivity. We're going to start this discussion in our next meeting here, but this is something deep that I want you to be aware of. I understand that we had just a few minutes to talk about it. I will certainly start with this because this needs to be reiterated and explained better. I didn't have time to do this, but we'll get to that, okay? Okay, bye then. ראיתי, לא, ראיתי, זה אני בסדר, כן, אני לא יודע בדיוק מתי זה גילה, באחד אבל אני מניח שנהיה באחת חזרה, משהו כזה, רק שיש את זה של יובל, אז אנחנו נמצא את הזמן. מה? כן, כן, כן.
זה pareil, c'est pas bon. Les deux? C'est théorique, c'est
io אני גם רוצה לעזור.
C'est juste après des mois, c'est
אני כאילו, זהו, אני מרגישה שאני לא ביטלתי אותו, אבל כזה פתאום קמתי בבוקר ומשהו קרה לי כזה, שנכנס למזכירות. כן, אפילו ברישום, לא, זה כאילו ישר היה עד הלילה, אבל אני נמצאת איך המזכירה בעניין. לא ידעתי לרישום אצלי, אבל אני גם יכולה לרישום ראשונה. אולי זה תואר ראשון. טוב, אני אצלנו המזכירות, אבל אני מסכימה איתך, אני לא רוצה, אני כאילו לא מביאה אז בשבילי לעשות את הקורס זה כאילו לעשות במקביל תואר אני לא יודעת, זה לא אמור להיות אבל אני לא יודעת כאילו ברמה שהוא דיבר תורלו ואני כזה מה זה
Yeah, it always scares my dog and we have to for her it's like shooting her in the head. Yeah, super frustrating. Every year we have to run after her super. No,
את הרעיון כאילו ולא בהכרח להביא חשבים אבל פשוט שאני אמשיך להגיע לשיעורים אבל בלי מחויבות
that and in school
באמת להבין את זה לא בלי חיים
לא מבינה איזה ישבתי על זה 3 שעות התקדמתי לדעתי אבל לא בגלל שזה קשה בגלל שהוא לא הסביר דברים מסוימים כמו שצריך אני צריכה שתהיה לי בזה הגדרות ואז אני יודעת לעבוד עם זה לא להסביר לא משנה בסדר אז שזה לוקח מלא זמן בסוף ממש אין לי זמן לזה כאילו
כן זהו אני ממש
כאילו קורס הזה אז גם אני לא חייבת אבל זה מעניינת כאילו כי רלוונטי אליי יותר ורציתי לשאול אם את בתוכנית כאילו פה של התואר נכון?
אני של תואר שני לא של
אוקיי אז יש לך מושג כאילו אם יש לא יודעת חייב או איזשהו מקום שאפשר להשיג ממנו מידע על הקורס איך קוראים
שאלה טובה צריך לשאול את החבר'ה מהאנטר
זהו, אני לא יודעת
נשאל את אילנה
Tu restes ici pour...
Ouais, au cas où, c'est des trucs intéressants.
Je pense que je vais aller lire plutôt...
Quoi?
Non mais le plus long du truc, la syndexique là.
Ah merde.
Dans le chapitre 1, il est bien fait.
Ah ou alors je vais commencer à taper peut-être.
Oui, ça. Et je taperai, je commencerai à taper pas une, parce que j'ai 14 heures à...